Awesome. All right. Well, hi, everyone. Welcome to an impromptu Systems Neuroscience and Complexity seminar. I'm really excited today to have Ryan with us, who's formerly of WashU and now over at the University of Washington, doing a joint postdoc with the Allen Brain Institute, which makes me deeply, deeply jealous of him because he's done some really fascinating work at this kind of cool intersection between statistical physics, neuroimaging, but also kind of embracing the role that the arousal system and the kind of natural way in which the brain's embedded within a biological organ. It's really embraced that, and I'm really happy to hear and see really cool work at that intersection. So really excited to see what you've been doing, Ryan. And if you want to take it away, go for it.
Yeah, well, thank you for that. I don't know if people, so I guess I don't know the back history of this, and if people, so I know you had reached out to me and said that it's kind of a journal club thing and we can just go through the paper figures, or we can.
That's perfectly fine. Yep. Whatever you'd like to do.
What people are.
Yeah, if you want to just walk through the figures and kind of walk us through what you were thinking, that's great. If you have other slides too, it's super cool, really casual. And I'd encourage people to kind of, as long as you're cool with it, Ryan, jump in and ask questions or push things in directions and make this as conversational as possible.
Yeah, exactly. And that's what I'm hoping for as well. So let me see if, let's see, this was, I think, is everyone here kind of familiar with resting state fMRI then? It seems to be the case, right?
Yeah, I think that's fair assumption. We'll butt in if we have questions about methodology or anything like that.
Yeah. Okay, well, oh, I guess I'll kind of start. I'll just pick up kind of where what seems like a logical spot for some background here. Can I share my screen here? So this work, if I can backtrack here. Well, so this work really came about as these are just some old slides that I kind of picked up just now, but this work really arose from recognizing these kind of parallel literatures going on in the study of intrinsic brain activity. We have resting state fMRI, this whole ordeal about brain networks and such, and then parallel line of work that was coming out in the rodent neurophysiology literature looking at brain states and the importance of arousal. And a lot of things, basically, you're measuring brain-wide spiking activity, calcium signals, that's this sort of thing, and everything seems to be very tightly locked to ongoing arousal fluctuations. And so what really struck us about this phenomenon was the time scale of these fluctuations, that they're fluctuating over tens of seconds, they're kind of dominating everything else, and this is the exact same time scale that we're looking at with resting state fMRI. We see these these slow fluctuations over tens of seconds in brain activity. And so, I guess to take this on, at this interface between brain states and brain networks, the question is there any relationship between these two things that kind of evolved in separate research communities, I would say. The second question that this work implicitly addresses then is, what is the functional role of this intrinsic brain activity? And I'll talk about some supporting evidence across species and modalities. Here, I'm just gonna focus on what was talked about in the paper. So that would be the human fMRI data and also some macaque electrophysiology. So to summarize this brain state literature very quickly, there's been in this awake behaving mouse setup, the head fixed setup, where you can study in vivo brain activity while the mouse is, performing some task, or can run on a wheel, that sort of thing. You can measure various forms of brain activity, shall we say, neural pixels recording thousands of neurons at the same time, calcium imaging, different metabolites. And you see that across all these measures, you have these large fluctuations in arousal that synchronize brain-wide activity. And I think to understand the role of intrinsic activity and arousal, and I guess going back here, I guess there's two questions that we can ask. One, first of all, why do we have these ongoing fluctuations in arousal? And two, why does everything in the brain, even starting at this level here, retinal output, why does everything seem to be so dependent on instantaneous arousal level? And so I think to do that, we have to go back to really fundamental biological principles. And this goes back to Claude Bernard and Walter Cannon and the idea of homeostasis, maintaining internal states against decay. And I think it's one thing to, to keep your internal state kind of clamped against decay, but then Charles Darwin comes along and says you also have to do this well, otherwise someone else is going to do this better than you, going to do it more efficiently. And so what this kind of amounts to is this efficient regulation of internal states of the brain and of the body, I should say. It leads to this predictive regulation of all these internal parameters of the body. So to give examples, some physiological variables that are tightly regulated, we have the overall chemical composition, temperature, blood pressure, all these sorts of things that are very tightly regulated. But as soon as we appreciate that this really needs to be done well, it's very inefficient to, have kind of this error-driven feedback approach where you're waiting for some perturbation to happen in some essential variable before you start correcting for it. So instead, what you tend to see is, the anticipation of a deviation of some internal variable. So for example, this little bunny venturing into a field, it's not going to wait until it's, starting to be eaten by a fox to say, oh, wait a minute, my internal states are decaying here. I got to do something about this. It's probably going to experience something that we like to call fear and probably try to avoid that situation altogether. And so that's kind of a form of predictive regulation of homeostasis here. Allostasis is the term people often use for this. And the second is that in order, again, to do this efficiently, you want to allocate limited energy resources according to anticipated needs. So, you don't, when you're in a kind of this fight or flight state and you're mobilizing energy resources to deal with that sort of threat, then there's other internal processes that you're kind of neglecting when you're doing that sort of thing. And so the idea is if you're able to allocate energetic resources according to predicted needs. So, that if there's a time where you can expect that you're more likely to encounter a threat, then you can kind of be more attuned to your external sensory environment and suspend more internally focused processes, for example, like digestive processes and that sort of thing. And so this tends to play out over these rest activity cycles, for example, sleep versus awake. In the most extreme case where, when you're in the awake state, you're expecting to deal with environmental behavioral challenges, that sort of thing, and you mobilize energy resources accordingly. And then in offline sleep states, of course, you can deal with more of the internal, in the brain memory plasticity, these types of processes. Energy resources can be more devoted to those types of things. So, this gets classically broken down into fight or flight, rest and digest, these modes of autonomic nervous system function. And we see that even during the awake state, there's this continual flipping back and forth between these states that synchronize essentially body-wide physiological regimes and also behavior and overall interaction with the environment. And then what we're starting to see now, we're contextualizing what I'm intending to contextualize by going through this background is the results from neuroscience showing that different dynamical regimes of brain function essentially are also varying according to these this cycle, this going back and forth between rest and digest fight or flight modes. So to focus on two ways in which these functional modes of the brain manifest, we have brain states, and this gets back to what I was just showing with all the rodent literature. And you can talk about different oscillations going along with different levels of arousal. For example, high arousal, gamma oscillations, or what's been referred to as desynchronized state, and low arousal delta waves, that sort of thing. So we can put that kind of on our map here, and you have high frequency desynchronized activity on the left and low frequency rhythms on the right here for low arousal states. And the idea in this paper was simply that brain networks also, we have a similar kind of concept of functional modes of the brain, and rest state imaging, we talk about these test positive and test negative networks that are really, kind of getting at the same idea of externally focused high arousal versus internally focused low arousal states. And so I guess to back up a little bit from that, we know that in addition to the temporal domain, brain function is spatially organized into functional modules with partially separable functionalities. And these things group into these large scale functional networks that we've given all these different special names. If so, we have these different large scale functional networks that the field has kind of converged on, roughly 6 or 7 major networks, spanning cortical and subcortical structures. And in addition to that, these networks, this is the whole hierarchy, starting with the Margulies et al. Paper, and obviously it has much longer history than that with Masilum and such, but this is the idea that these brain networks, these canonical brain networks, can be placed on this principal axis, moving from early sensory motor regions to higher order association regions in the brain. And the idea is simply that different sets of regions, you can kind of, so take, for example, the default mode network. It's a distributed network with frontal, parietal, and temporal regions, and yet you can kind of group them all along this axis. They all have they occupy similar distances from early sensory motor regions. So the unifying principle of the default mode network here is that it comprises regions that are furthest away from early sensory processing in the brain. And you can do this for all these classical networks. So this kind of gives an alternative view of how to understand functional organization, large-scale functional organization across the brain, is this variation along this principal axis going from sensory motor to association regions. And the key idea here is that if you think about what this is really dividing up here, it's another form of this external to internal. So this is what I was referring to earlier, this idea between activity and rest. So the idea is that this rest activity cycle is also spatially embedded along this principal axis of brain functional organization. So the idea now is that, again, in addition to going through these body-wide and behavioral states corresponding to rest and activity, these are also associated with different spatial patterns of excitability on the brain that correspond to these large-scale networks. So, for example, here we have this idea of a task-positive set of networks in the brain that are more focused, more related to sensory motor activity, the immediate sensory environment and such. And then, for example, the default mode network that is more internally focused. And so the idea here that's really, or I guess the kind of the theoretical framework underlying the analyses pursued in the paper was based on this idea of a spatiotemporal regulatory process for brain function here, where you have this ongoing arousal fluctuation, arousal cycle. You're constantly evolving along this cycle, and resting state imaging is really capturing this process. And because things are, let's see what I think I can skip all this stuff that we have the phenomenon of resting state functional connectivity, the idea that intrinsic activity has these patterns of zero lag correlation within these large scale networks. And we can recover the topography of these large scale functional networks on the basis of zero lag correlation between regions in these spontaneous fMRI signal fluctuations. That has been a very productive area of research, and still lacks kind of a unifying framework for thinking about the functional contribution of these fluctuations, the physiology underlying them, and in particular the phenomenology of them. It's really become an area where a suite of different spatiotemporal analyses are applied with. It's clear that they have a lot of connections to them, but there isn't really an agreed upon way to think about all these different dynamical processes going on, and the simplest analyses that pull out the structure and the basic the essential aspects of what's really underlying them. So the idea here is that we're putting forward in this paper is that these ongoing arousal fluctuations actually synchronize brain-wide activity as we expect from kind of the mouse, the rodent neurophysiology literature, but that different phases along this effectively oscillation, along this rhythm, are associated with different spatial patterns. And these spatial patterns correspond to the resting state networks that we know and love. But again, because this rest activity gradient, this principal coordinate, because it's embedded as a spatial gradient across the brain, moving from these sensory motor to association regions, ultimately to the default mode network, the idea is that this is actually, this spatiotemporal regulation is going to play out as a traveling wave on the brain. As you advance kind of along this rest activity cycle, this arousal cycle, activity is going to slowly propagate from these early sensory motor regions along this principal coordinate to these association regions and ultimately the default mode network. So does this all make sense so far? Just whether or not you agree with it, I guess does what I'm saying make sense, and the basic idea. This is kind of the conceptual framework more independently of resting state fMRI for how we're thinking about the physiology and function of these fluctuations. And then the paper became basically a suite of analyses that were kind of testing predictions of this framework as kind of a step forward in unifying a lot of the analyses and electrophysiological and behavioral correlates that have so far been discussed in relation to resting state fMRI fluctuations.
Yeah, so does anyone have any questions? Any students want to ask anything clarifying? It was a really nice introduction to the kind of broader concept. I think one thing that's kind of interesting to think about is that a lot of times when we conduct the kinds of analyses we do of resting state, we kind of ignore phase. We kind of look at, you know, a 10-minute block of someone in a particular state versus some other state, anesthetized or not, or this clinical group or that clinical group. And so in a way, it kind of, there's been all this hidden information or potential information in resting state data in particular, but involved in general, that's kind of been kind of systematically ignored for a long time. And you know, maybe you'll get to this later, but I was really curious how what you found here relates to things like what Anish Mitra found with Mark and colleagues with those lag threads, and whether or not they're kind of a different way to look at it. But
yeah, that's a good question. I think that that among in addition to a lot of other processes people have described, my view on that is that it's something that is likely to emerge if you start with something as simple as this process going on, and then you do all your standard steps. You do things like global signal regression, regress this, regress that, do any type of spatiotemporal decomposition. And the point that I want to get across there is that if you have, so basically there's no good way to spatiotemporally decompose traveling waves. And the reason being there's spatial dependence and there's temporal dependence, and all these different algorithms for spatiotemporal decomposition typically assume some you know, that things are either separable in the spatial or temporal domain. And so take, for example, something like temporal ICA or PCA. Lags are enough to, you know, if you take a signal and time shift it and you have a lag copy of that signal and you do some type of dimensionality reduction, it's going to tell you that there are two kind of independent components, even though, you know, we can understand that we can understand the rank to be lower than when we think about this just being kind of the same process, but just lagged in time. And so that's basically, yeah, maybe I can just pull up the paper actually. So this is kind of where, this is the basic conceptual model for how we're thinking that this plays out, is again, that you have this process on this always ongoing process. You're kind of moving along this cycle constantly. Temporally, this manifests as these ongoing arousal fluctuations over tens of seconds, the same time scale that we see resting state fMRI signal fluctuations. If we want to look at in-phase relationships, so this is zero lag correlation, that's how functional connectivity is usually defined, is through zero lag relationships. The idea is that these traveling wave, I guess I'll keep flipping back and forth here because I like this picture better. We see that instantaneous activation pattern, so to speak, the spatial pattern on the brain, those are going to be very topographically organized. And so that kind of gives you your resting state networks at zero lag. The idea is that even though you can get these distinct looking patterns at zero by using zero lag correlation measures, they're all synchronized with one another and synchronized to this global arousal fluctuation. They're just simply phase shifted from one another. So the idea is so more generally, so Mac, you brought up lag threads, and I would say more generally to this account is intended to kind of unify zero lag functional connectivity with this rich literature describing spatiotemporal dynamics in the signal and propagation structure and that sort of thing. And the idea is that you have a propagating structure, this traveling wave structure that gives rise to topographically organized patterns of zero lag activity. And that is what we capture on the one hand with functional connectivity. But if we were to use lagged measures of correlations and that sort of thing, that's how we see this, uncover this rich spatiotemporal structure. So that's the basic idea behind this model. Yeah, Kale?
I have a question. So I feel like it's kind of unfortunate that so much functional connectivity started with resting state, where there's nothing really special about staring at crosshairs. And I'm curious what you might think about if somebody in the fMRI were looking at a series of pictures, and would you expect this same phase shift between more visual motor areas, then mind wandering to default areas and back and forth for a task that was movie watching or series of pictures, yeah, joystick tracking and back, what have you, because functional connectivity, zero lag functional connectivity generally shows the same networks. If you're not analyzing a resting state scan, but a task scan, it's irrelevant. So I'm curious how the phase and modes work.
Yeah, this is a good question. This is something that I think about a lot. It's and I've only done some very preliminary analyses on this, but they do seem in line with the perspective that basically this intrinsic process is always ongoing. There's a lot of literature on task fMRI scans that you have kind of this signature, like an onset transient. And I kind of conceptualize that as like a phase resetting effectively of this arousal process. This appears to be, it's ongoing during tasks and such. And then I believe task responses are kind of, you could evoked responses, evoked activity, those are kind of superposed, maybe not linearly, but superposed on top of this ongoing process. But this definitely doesn't go away when doing a task in the scanner. Does that answer the question?
Yeah, I guess my question is it synchronized? It seems to be like there's an independent phases. You're suggesting that the visuomotor would be on, and then a few seconds later, 30 seconds later, would be more default mode,
sure,
regardless of what the person was actually doing in the scanner. It's like there's two separate trains going on. Is that what you're saying? Or are they synchronized? Is the arousal to internal synchronized with the task?
I think it is. That's a good question. I think it is, and I think it's I don't think they're independent by any means. I think and to make this more kind of, to get an intuitive appreciation, I guess, I think in the task literature, this is what's referred to as kind of, this connects to the performance variability literature and, the spatial correlates of enhanced versus decremented task performance and that sort of thing. And, you know, and even, you know, Mac, you've talked about segregated and integrated states of the brain and related that to pupil size and behavior and such. And this is all, I think, they're all different languages, different perspectives on the same basic process going on. This is just how I'm kind of conceptualizing this. And so you know, think about, so you enter, so, you know, a key, very reproducible task result is this turning on, so to speak, of this task positive network, hence its name, and a deactivation, so to speak, of the default mode network. That if we kind of put that on this cycle, that's exactly what we're saying, is that but it's just a little bit more subtle. It's not necessarily like, it's not just a deactivation, it's anti-phase, it's out of phase. And so, you know, the start of the task period is associated with, you know, for example, this wave being in the position of the task positive network, and it's out of phase in areas of the default mode network. And you could think about it as, you could ask, you know, why can't we sustain our attention indefinitely? Why can we not keep our default mode network turned off indefinitely? And, you know, I think it's quite plausible that shifting of that eventual mind wandering process, the lapse in attention, is going to be associated with, you know, eventual coming, but, you know, this rotating around and going towards the default mode network. So I think they're highly intertwined. They're not independent processes at all.
So Ryan, were you thinking that the kind of loop that you've been describing, this kind of like bounce back and forth between sympathetic and parasympathetic or something like that, is kind of has a characteristic sort of frequency, or is it the kind of thing that can change? Because it almost seems like during certain contexts, having this spatiotemporal traveling wave could be problematic. Like if I wanted to remember a set of items or something over a delay, if my brain starts rolling away from me and I start mind wandering, I'm in big trouble. And so it's about being able to hold that in a steady state, like a standing wave. I'm really only bringing this up to tease Kevin Aquino, because I think standing waves are more interesting than traveling waves. At least we have that.
Hey, I think this,
I'm just curious what you think.
This is a good question. I'm very glad you brought this up. I have two answers. The first is more of a, the first is to reiterate that I don't think we can kind of freeze the traveling wave in a certain spot, there's limits to the extent that we can do that. And that is kind of directly related to our inability, our subjective experience, the inability to sustain our attention indefinitely. It's going to be associated with decrements in task performance. And I think the reason I went about this kind of roundabout background in setting this up is because, you know, basically, you know, this thing it didn't evolve to, you know, perform well on the types of tasks that we're doing in cognitive neuroscience indefinitely. You know, eventually you need to turn to like making sure that you have enough energy resources and that you're digesting and, you know, all these sorts of things. So, I think you're right that it's going to result in decrements in task performance, and that's not necessarily a bad thing from more of an evolutionary perspective. The second point, you bring up the frequency, and this is maybe a good time to jump into this result here. So to one of the predictions that we go through in the paper is the idea that bold signal fluctuation should be globally coherent with arousal. And so to do this, we just averaged fMRI signals within each of these large scale networks and looked at temporal coherence. So this is correlation as a function of frequency across the canonical range of fMRI signal fluctuations. So order of magnitude, 0.01 hertz to 0.1 hertz. And the idea is that the prediction is that you're going to have a strong coherence across this frequency range. And that's what we see. And you see that there's a peak here around lower frequencies, around 0.025 hertz. And that connects with some older literature on the so-called infraslow oscillation. But more generally, I should say that there's a bias. There seems to be, so for people thinking in dynamical systems in this group that you mentioned, there seems to be some limit cycle that it does evolve towards, but this seems to be a pretty flexible thing. People have referred to it. The Palvas have referred to it, and Shella Kielholtz's group, they've referred to this as quasi-periodic. The idea that there is some rhythmicity to this thing. It has a dominant frequency at this low frequency at this specific time scale, but at the same time, it seems to be a very broadband phenomenon. During the awake state, it seems to be constant or continually phase reset. And so there is a lot of flexibility in that time scale. There's a bit of, if we look at the phase relationships between networks, this was another key prediction here, is that all brain networks are synchronized. Their magnitude of coherence is going to be statistically significant with ongoing arousal fluctuations. We're indexing arousal through respiratory variation, just measured from chest belt data. And the prediction, though, is that the temporal aspect of coherence, so the phase of coherence, is going to have topographic specificity. And that's what we see here. So basically, these blue and purple motor and visual networks, they're becoming active first in their temporal relationship to arousal fluctuations. And then you see these other networks kind of following in line. I'm trying to get my pointer. Oh, here we go. Then you see these other networks falling in line. And you can do this with various measures of arousal. This comes out the point that I'm about to make, it's illustrated best. It holds up quantitatively for other measures, but it's illustrated best here for pupil size. Basically, this flat phase relationship. So networks have these phase shifts relative to one another in their relationship to ongoing arousal fluctuations. The consistency of this phase shift across this frequency range, this broad frequency range, that is actually that's highly non-trivial. It's actually highly non-linear. So if there was just, this is another point of connection to all the work that like Rakel group has been doing on propagation and time lags. If there was just a time delay between two brain regions, you would expect that time delay to scale linearly with frequency. You would expect the phase shift to scale linearly with frequency. So to put this, to give an example, if you have so here we're talking about the difference between, let's say, a 20-second or 10-second oscillation, I'm getting close to that, and a 100-second oscillation. So, if you have a time delay of 5 seconds, then that's 1/2 the period of a 10-second oscillation. So that's pi. That's a phase shift of pi. But then that same time delay, 5 seconds, is now 1/20th the phase shift of a 100-second oscillation, right? So that's 2 pi over 20. So that's a very small phase shift now. So basically, you should see that these phases all come together. Phase shifts start out very big and all kind of come together as they move towards zero. And that would be consistent with the linear time delay between brain regions. It turns out that this is much flatter, even for respiratory variation. This scales much more, it's much flatter than we would expect for a linear time delay process. And that's consistent with this idea of this cycle that that's constantly playing out. There's an oscillation, but it's a broadband, it has a broadband characteristic to it. So it's constantly, you're constantly evolving along the cycle, but from cycle to cycle, you're changing in the duration of that cycle, how long it takes. So you end up with this very broadband frequency context. There isn't a clear 20-second oscillation, but the phase relationships between brain regions remain intact, and that's the key point of this cycle. So that was kind of a
awesome. Thank you.
long answer, but does that make sense, Matt?
Really good answer. Thank you. I think Kevo had a question. Do you want to jump in?
Yeah, just kind of sort of related. Well, 2 questions. The first off is, does it make sense to instead look at the echo after some tasks? So, in this analysis, would it make sense to instead have an experiment where you would force someone to do some particular cycle that you would like, for example, follow the same cycle, have the same kind of trajectory from different networks, stop the task and then look at the echo, so to speak. And so then you could constrain whether you go into mind wandering, that kind of stuff.
Yeah, I mean, yes, it's a brilliant suggestion, brilliant idea. I ran out of time to do these things. I would love to. I think there's a lot of interesting follow-ups to this sort of thing, and that is certainly one of them. Basically, if I understand correctly, you're getting at basically testing the entrainment idea, right?
Yeah.
And seeing if this kind of follows through after. And to some extent, there is literature on this. So, I like to kind of describe this work as really what it was I kind of motivated from a different perspective today, but really what it was an attempt to kind of unify a lot of the neuroimaging literature, a lot of the findings in the neuroimaging literature that have kind of existed in kind of a scattered sense. And, this is one of those findings that fits within this framework, the finding that if doing it like at the offset of a task block, default mode network activity ramps back up pretty quickly. And that seems to fit right in line with this idea that, you're phase resetting the default mode network essentially during task performance. And so in fact, we do already know that, that once the task block is complete, you do see, no one's explicitly quantified in terms of propagation, but we do see this ramping up of default mode network activity after the task. So, yeah.
Well, I'd be interested to see, if you have a task, you'd stop, ramp up to default mode network, and then I guess vary the time you start a new task. If that corresponds to behavior, depends in phase of where you are in the cycle.
Yeah. I think that'd be so cool. Yeah.
I think
That's a great idea.
Garfinkel has done some work of like the relationships between, heart rate and performance and how that changes, if you're synchronized to your heartbeat, you have differences in fear, all that kind of stuff. Then maybe there's some sort of global network you're wondering that involves brain and also body, and that kind of independence way around wave cycle.
Yeah, that's interesting. I think you could definitely do something like that. And again, it's a lot of these things, they haven't been explicitly mapped to this cycle, obviously. But this does, I think, connect to, there is a lot of literature on, as task performance and relating that to different patterns of instantaneous, bold signal pattern across the brain, or functional connectivity states and that sort of thing. And right, you could conceive that you could now maybe go back to some of that literature and say, okay, well, could we maybe more rigorously tie these results into, or explicitly link them to phase of the underlying underlying arousal cycle in these various task conditions. Yeah, I think that'd be a very interesting extension of this.
Another question I have just on the concept of phase as well. I always find it hard to trust phase in bold because bold is slow and all that sort of stuff. In terms of how do you control? Because you have the phase of this electrophysiology and you have this convolution, I guess, signal speak from neuroactivity to bold. And so in some ways you're kind of assuming that the convolution is the same, I guess, brain wide.
Yes.
So, how do you deal with that in your mind and sort of...
Yeah, I'm glad you brought this up. I think this is an important clarification for the present work and really, I guess, the ideological approach here. So the idea here, and this is partly why I kind of went to, motivated from the direction that I did, the standard conceptualization of fMRI, standard model of fMRI, so to speak, is the idea that you have fast neural processes that are transduced through this slow hemodynamic response. And that kind of conceptualization evolved, of course, through the test neuroimaging literature and such. This is motivated from this perspective that there is a bona fide so-called infraslow physiological process. It is actually occurring on this time scale. And this is motivated, some of the mouse literature that I was talking about, calcium imaging, firing rates, that sort of thing. All of these indices of brain activity are fluctuating on this so-called hemodynamic timescale. So the neural measures of interest too are also fluctuating on this very slow timescale. So we're interested in this slow timescale process, and that kind of gets us past this an additional modeling step of saying, what does the hemodynamic response look like in this region, that region, et cetera, and convolving and that sort of thing. I didn't do any type of convolution. I just treated the fMRI signals as if that is the physiological process of interest. And so, right, so everything we're looking at is at this very slow time scale, but you can do this sort of thing with non-hemodynamic measures of neural activity and get the same result.
Mm. Yeah, I mean, because I guess it's I always find it quite interesting, because I guess when you look at tasks related in terms of your energy reserves, and you're dealing with resting state, you're dealing with sort of different ATP cycle, and with tasks you have an increase in blood flow, and that changes the hemodynamic response. It makes sense to keep it all within the same kind of energetic process of this sort of in resting state. Yeah, I mean, my background's in modeling hemodynamic responses, so I get this question all the time when I do anything like this.
Yeah, well, it's interesting. So even task responses, it seems that there's very fast spiking activity in response to the task. But then if you were to look at broadband gamma, for example, gamma frequency range, you'll see this slow response on the time scale of tens of seconds that very tightly correlates with the bold signal. And so the perspective here is that that's the physiological process that we're looking at here, rather than kind of this low pass filtered washed out version of fast millisecond time scale neural activity. Yeah.
I had a question about the task delay for different networks and how that might influence the phase issue. If I don't know if people have looked at how long it takes. You know how we think of 2 to 5 seconds for bold from.
Oh, this is interesting.
If default mode is like 2 seconds and visual is 5 seconds, then that might be a problem. But
this is a very interesting question. The most rigorous, the reason this is interesting is because the most rigorous analysis of this that I know was done recently. Someone might be able to help me on the first author's name. I'm blanking on it right now. It was published in Neuroimage a couple of years ago. And they estimated all these different parameters like full with half max and delay and such of the hemodynamic response.
I think it was Dan Marinazzo's group, right?
Yeah.
Oh, was it?
Yeah,
okay. Yeah. Basically, if you look at time to peak of the hemodynamic response, you get out the same topography. And I think there's two perspectives on that. One is that that is truly the hemodynamic response time across these different, it varies according to this logic. Or the alternative possibility, which I tend to favor, is that when you're looking at task responses, you're kind of looking at the same, again, the phase resetting of this process, that sort of thing. And so, basically it just takes longer to maybe phase reset to the default mode network, for example, something like that. So I think, but these are, what is known is that, like the conclusion in that paper was that you have some regional variability in hemodynamic response times, and that topography largely recapitulates this. And so that would have actually, I think, been a major concern about the interpretation of these results, if not for also, the electrophysiology that we included in this paper showing that you see a similar regional delay structure in spontaneous activity. So yeah, that's a good question. I think, so it definitely complicates how you would, if you now, knowing that this manifests in electrophysiology, if you now revisit the resting state or task hemodynamic response literature, I think it does become a challenge to say, what is the delay of the hemodynamic response versus what is just this region being slow to respond, just the brain activity in general. And I think that's the sort of thing that requires simultaneous requires looking at things across both electrophysiology and fMRI.
Can I ask a quick question?
Yeah, please.
So just to understand, maybe there's another way to frame this phase issue. I like to just sort of make everything sort of really mechanistic. So if you have these regions that are sort of bolted to different phases, if we sort of translate that into the time domain, since the phase relationships are a little bit, like the phase coherence length is not very long, like you said, it resets. So is it the case that you're kind of stretching, like deforming the entire time series of oscillators in a nonlinear way?
Yes.
So, and that leads to the question, if you were to look very closely at a moment of phase reset, is it something like a sort of phase speed up, as opposed to, like, if you're simulating something, you could just have a hard phase reset, right? So
I don't know.
Okay.
Yeah, that's a great question. I honestly, I haven't done a thorough analysis of the phase resetting process. That part is more kind of more hypothesized in order to be consistent, with the existing literature. We kind of presume that there is some phase resetting type process going on. I think it'd be fascinating to know whether, it's a hard reset or you basically very quickly kind of cycle through to the next position. That's a fascinating question, actually. I don't know.
Because what you just said sounded more like quickly cycling through.
Right. Right. And
it seems to make a little sense if this relationship is so reliable. And I think I couldn't necessarily work this out, but what you said about the relation between this and the HRF might actually fit in with this idea, which is that it takes a little longer to get to where you need to be, and there's only so much you can do to like truncate that, and that varies from region to region.
And that is what I had in mind when I gave the answer. But you're right. That's a total open question. So that's very interesting. This is the most I think I've thought about that distinction. So I think that would be very interesting to follow up on. Yeah, thank you for that.
Thanks.
Do we have any other questions at this stage? Have we completely derailed have we like phase resetted your cycle so much that?
No, I mean, this is kind of what I intended. I can kind of just keep stepping through the figures, but I really just meant to kind of, get it up to the point where the framework is introduced and such. And I think I was expecting most of the questions there. And, the analyses I think are kind of straightforward from there. So, but we can kind of step through them and see if any other questions come up.
So one of the things that I liked a lot about your paper, but one of the things I liked was that you tracked subcortical structures as well, and you found kind of really compelling patterns of kind of phase relationships there. Do you want to? Does it make sense to maybe jump to that point, and then we can, because we've done a little bit of work thinking about, say, the thalamus and its relationship to the cortex, and
I'd love to get your thoughts
on connectivity profiles.
Yeah. Actually, I think that's the next slide. Yeah.
Oh, perfect.
So this is just still at the coherence level. So this isn't looking at it, actually displayed on the brain, but we can repeat the same procedure where we just average the signal within each of these topographically defined regions, these networks, but the network representations within each of these subcortical structures in cerebellum. And we can kind of focus on the networks that are actually represented in each of these structures. And there's 4 major ones here: motor in blue, default mode in red, singulopercular, attention related kind of E network, and frontoparietal control or executive control in orange and yellow. But again, we see coherence across this broadband infraslow frequency range, and we see this peculiar phase shift pattern across each of these structures, where you have blue and purple, so motor and singulopercular control networks, so kind of task positive regions. Down at the bottom, those are earliest, and then latest we have default mode and frontoparietal, kind of this task negative system. And yeah, it comes out pretty cleanly across these structures. Striatum, it's kind of squished, and that's likely a noise issue, reduced SNR. And so you have a lot of synchronized noise that kind of compresses things. But
you're also not really having the participants do much of anything, and the striatum is really heavily involved in kind of action, sort of,
yeah, that's an
selection's the right
point.
word, but yeah, you can imagine if you put people in a particular context, all of a sudden you'd have more dopamine flowing around, you'd have more cortical inputs, driving the strider. So you might see something a bit more interesting there.
Yeah, that's a good point. Yeah, so we see this topographic sequence playing out across these structures, and I think I'll flip back to the paper now. So if we want to look at this now across the brain, what we can do is, so here we're looking at coherence as a function of frequency. And now if we want to look at this across the brain, instead of dimensionality reducing according to these networks, we can look at it across the brain, but focus on a specific frequency. So we've already shown that this exists across all these frequencies. Here, we're just going to filter into this frequency of maximal coherence, this, around 0.025 hertz-ish, and look at the phase topography across the brain to see what that looks like. And this is just illustrating that procedure using Hilbert transform. But using time average phase relationships, we get out this map. And you see this blue to red, so early to late organization going from, again, these earlier sensory motor regions. Primary visual cortex is kind of an interesting outlier there, but moving towards transmodal association regions in, for example, the default mode network, regions of the default mode network here. And you see the same thing playing out in subcortical structures. And to kind of demonstrate this quantitatively, we use the same dimensionality reduction, nonlinear dimensionality reduction approach, the diffusion maps, for obtaining this principal functional coordinate from the functional connectivity data across cortex. So this is the Margulies et al. Result that I think a lot of people will be familiar with here, this principal gradient idea. You can do the, apply the same analysis to subcortical to cortical functional connectivity, and your principal eigenvector, I should say, across these different structures gives you a similar sensory motor to association axis. So here, for example, ventral nuclei, lateral nuclei moving toward medial and anterior regions of the thalamus. And then similarly in striatum and cerebellum. And then we can just basically correlate the rank ordering of brain regions, brain voxels, according to their phase versus according to their position along this coordinate. And the idea is that those would be very correlated. And the claim here is that, the most parsimonious interpretation of this is that, in fact, this wave process gives rise to these this structure in the data that we obtain with functional connectivity and decomposition in functional connectivity matrix. And that's tested here. And F's functional connectivity rank, so principal coordinate rank along this vertical axis, and then early to late rank along this horizontal axis. And we see pretty strong correlations across each of these structures. So in general, there's a tight correspondence between this wave propagation process as assessed by phase relationships and spontaneous activity, and the principal functional connectivity coordinate obtained through diffusion method embedding.
Yeah, it's interesting that I don't know if there's something to this in terms of the kind of different signals that you'll get out, but the thalamic relationship to phase looks really clean, but then the cortex looks really gnarly and non-linear. Like there's a kind of general trend from the bottom left to the top right, but there's a whole bunch of regions that really aren't following that pattern. And like in particular, like there's regions that are the same color, but are in very different parts. So like this group of purple versus that group of purple. It's interesting.
Oh, that should, so I think, so in general, the colors clustering together, that reflects basically functional connectivity networks, right? So that just means that similar regions that belong to the same network will be clustered together on the horizontal axis because they have similar phase, and that's why they have strong functional connectivity, because they have similar phase. And then they'll be clustered together on the vertical axis because they occupy similar positions along this functional connectivity.
On the Margulies gradient. Yeah.
Exactly. And then
so what do you then make of the smearing of phase in otherwise what we would think of as coherent?
Yeah, this is, I think everything else it actually follows pretty nicely along this coordinate. Light purple is the singular percolate network. So to be clear, different from the the visual. This is the outlier here, right? This is odd. This is very interesting, and you know, actually, I'd love it if anyone had any ideas on this, any interpretation of that. But all I can say is that it's actually quite in line with observations from resting state fMRI and functional connectivity. So you see a very strong distinction between primary visual cortex and higher order visual cortex and functional connectivity. You see, and that's basically reflecting like an eccentricity organization of the occipital cortex. You see that when you close your eyes and presumably have less, you know, during the normal resting state, the argument, at least in this paper, is that this is the predominant physiological process, and you also, of course, have intrinsic physiological process, and then you, of course, also have like, you know, moment to moment evoked activity and, you know, what you're thinking about or whatever, and that has some contribution, of course, to the signals. But when you close your eyes, presumably this sort of thing is dominating the visual cortex activity, and when you close your eyes, V1 becomes especially distinguished from the surrounding visual cortex in terms of its functional connectivity. It also starts to couple more with motor cortex, which is what we see in the wave process, which is quite interesting. So I don't know how to interpret the fact that early visual cortex seems to be quite late in this result, but it's actually very consistent with resting state observations more generally. So
is the prediction that if someone's eyes were open, that that pattern should be much more aligned with the kind of tealy, light greeny color, or?
No, because we're time locking this process to arousal. So this is after a lot of averaging and such anyway. But in general, yeah. So I think this is kind of what that it does in fact propagate, you know, from higher order visual cortex to the occipital pole here. But I think if you're measuring functional connectivity, for example, then you're going to have much more general synchron. Like the phase shift right now is huge between higher order and visual cortex and early visual cortex. But during the awake state, when you're actively seeing things, et cetera, and have lots of evoked activity, there's much stronger synchronization across all visual areas. And so, you know, when you do functional connectivity decomposition, you generally tend to get a monolithic visual network, and it doesn't separate out into like early visual cortex and higher order visual cortex until lower levels or finer levels of the hierarchy, I guess.
You could look at, I mean, if you overlay like an eccentricity map, I mean, fovea is huge in V1.
Oh, this looks like a, yeah. This looks very much like the eccentricity map. There's papers dating back, even, you know, actually, I had to I rediscovered this actually in the Thomas Yeo Kreenan, you know, 2011 paper. If you go back and look at that, actually, they show images of this eccentricity organization of visual cortex according to functional connectivity. And there you see these principles where you have both. So if I zoom in, oh God, that's so pixelated. If I zoom in here, you see two organizational features. One, you see a general progression from, you see the eccentricity organization going from blue to here, reddish at the pole. And then you also see a strong outline of presumably striate cortex, V1. Both of those principles are actually discussed in the Yeo et al. Paper. They stumbled to both of those features. So
I think it's also in the Glasser 2016 paper. I think it's
also in the Glassers, yeah.
Yeah, talk about it.
Yeah.
I remember that figure.
You're right. And it's, you know, the Steve Peterson, Goggenwig, Tim Lauman, that group, all studying parcellation according to functional connectivity, which is, you know, what the Glasser paper did. That was kind of their strongest argument that they're getting at areal boundaries. Is that was their canonical example. They'd be like, look at V1, you see the sharp distinction. So it's interesting that it recapitulates those properties. And so to the extent that, you know, the argument is that this is explaining a lot of previously observed features of functional connectivity, I think it holds up. But then the physiological functional interpretation, I think, is much more open. Yeah.
I have a general question about these waves moving through, how does that correspond with dynamic functional connectivity sliding window approaches, where they identify individual states of functional connectivity as opposed to looking at phase?
Yeah, my interpretation of a lot of that literature is that you can get out. There's been a few papers now actually that have strongly linked, and Mac, this ties into your previous work too, that have strongly linked dynamic functional connectivity states to arousal. Basically that different.
Yeah, Todd's Constable's group just had a really nice one, and there's another one as well that we had. Ted Satterthwaite and Danny. Who was that guy that he joined us? He was really great. Nick. I'm completely blanking on his name. Anyway, yes, there's been tons of really interesting stuff recently.
Yeah. So it's actually a very tight relationship, and so my interpretation of this, I didn't show this explicitly, but is that when you start to talk about these cycles kind of change, the duration changing from, and other statistical properties of these cycles, their amplitude and that sort of thing, changing from instance to instance of this cycle. That's the type of thing that I believe, one, reflects slower changes in arousal, vigilance, that sort of thing. So you have this ongoing cycling with a relatively strict time scale of tens of seconds. And then, let's say you're just sleepy one day, or you're, you're drifting off on a time scale of several minutes, or that sort of thing. Those things I think are reflected in kind of the second order properties of these cycles. So for example, duration, amplitude, and that's the type of thing that seems to be reflected in dynamic functional connectivity. I think the most important thing there is that there is a converging literature that dynamic functional connectivity states seem to be intimately linked to slow fluctuations in vigilance.
We had a couple people that apologized they had to leave, Ryan. Alex and Kevin both had to go, unfortunately, but they'll maybe catch up with you later.
Thank you for your talk, Randall. Sorry.
Oh, Kevin's
yeah, no, I appreciate it.
Classic Aquino lurking. Oh, I've left everyone.
I'm just saying, see what you said about. Right,
hey, thanks for coming.
No worries.
Do we have other questions? I kind of wanted to come back a little bit to the kind of functional interpretation here in a way, and it's always, I've always sort of, ever since reading like Bayou, He's papers with Marcus back in the day, there's this kind of interesting kind of realization that this really infra-slow feature of these signals is kind of inconsistent with the sort of speed at which we feel, at least subjectively, that our cognition evolves. We have this kind of, there's this really interesting relationship, this kind of like 300 millisecond time scale where you get this ignition event and you're aware of a thing, and then there's this thing in the background that's kind of like slowly moving along, like and
Agreed.
I've often found that those things are kind of really hard to reconcile, and I just wondered if you had thoughts about that. I've also seen Claire's raised her hand, so feel free to give me a short answer, and then we can see if Claire has a better question.
Okay.
We kind of can, it's a similar kind of question.
Oh, okay. Go ahead.
So I don't, maybe I can kind of jump in, because it's basically a very kind of similar question. A little side point. Oh wait, no, I'll leave the side point. But yeah, I kind of wanted to ask a similar thing, sort of what Max said, and your paper, and also this kind of this other work that was that I wanted to mention. It was kind of in relation to what Kevin said, where he was talking about Sarah Garfinkel's work, where she sort of tried to look at the relationship between heart rate variability and different sort of cognitive states. There's this really awesome work by Catherine Talon-Baudry. I don't know if you've heard of her. Yeah, she's, I think, in Paris, and I saw her present on it a few years ago, and it just really amazed me, the kind of questions she was looking at, because she's been looking at the effect of sort of rhythms from heart rate variability, but also gastric rhythms and the extent to which they're driving resting state, right? And she did some kind of like amazing work where in the resting state, like 10% of the variability or something is controlled by our tummy, which is crazy. But she did some, she did. She has a really beautiful study where she tried to sort of tie in, I think it was with heart rate variability, but sort of tried to tie in, I guess, the sort of, I guess, cognitive ramifications. So whether or not you're kind of surfing the wave with these intrinsic rhythms. And I think they found that when there was sort of more coupling between heart rate rhythms and these infraflow brain rhythms, that people were much more inclined to be mind wandering in a sort of self-related way. So it was kind of driving this more self sort of directed processing, which I think is a kind of neat thing. And that was thinking about that was sort of got me to the same question of Mac, and yeah, I wanted to sort of ask you sort of how we're supposed to think about that, because so I kind of think about these more kind of quick, I guess, cognitive type actions that sort of determine on what scale we're thinking and feeling. If you kind of imagine it like this sort of herd of porpoises swimming in the ocean, right? So you've got these kind of active porpoises can kind of do their own thing, but then locally they're working within a current, right? The current of the kind of waves around them, and then more broadly you've got the tide, right? So you've got these things kind of happening at different levels, which they constrain each other, but they're free of each other to a kind of extent. And I guess I wanted to understand if the stuff that you're pointing out here with these sort of infraflow like phase shift stuff is the way to think about it, that we have these more kind of quicker cognitive thought type things, that something that's sort of overlaid on top of those, and those are happening sort of at another level beneath, and maybe even sort of slow ones, and the way that they might kind of help constrain, I guess, more I guess quicker sort of thought things is sometimes they sort of work together and help shape each other. Maybe sometimes they're a bit sort of antagonistic, and maybe there's this sort of adaptive sweet spot where these are kind of aligned. And I guess that's kind of what brought me back to Catherine Pallo and Bodji's work, where when these rhythms were aligned, it shaped a particular type of thought. Yeah, it was very badly, but I think it kind of gets you to the same point. No, actually, I talked about porpoises, so. Right, that was better.
No, I love the metaphor. I was just gonna say, Mac, does that answer your question? Are there any more questions? I'm serious. I mean, I think that's I really clear. I think that's exactly how I conceptualize it. I think that's spot on. And if you go back through history, I mean, this isn't just neuroimaging and infraflow, but this goes into the same idea that people like Gary Bizaki and before him and Mircea Steriad and people have been talking about, this hierarchy of rhythms, for example, right? This nested oscillations, cross-frequency coupling, that whole idea. And basically, I think we have both an intuitive and an experimental bias toward looking at fast timescales and thinking of cognition in terms of fast processes. And I think the intuition there is basically, we can think very fast, therefore that's the timescale of brain activity. And in reality, I think, if we just look at, if we take an empirical approach, I mean, if we just look at the power spectrum, for example, these brain signals, you mentioned, Mac, you invoked BU's work that's about scale-free 1/f scaling of brain activity. And what that means is you have exponentially greater power in these lower frequencies in your signal. So these very large fluctuations in the signal occurring over increasingly slow timescales. If you think about, I'm saying things very quickly right now, but there are many things that are changing much more slowly in my mental state right now, my hunger state, my
you're thinking about porpoises riding around waves in the ocean, aren't you?
I am exactly.
I'm wondering what the collective noun for porpoises is. I went with herd, but I think it may be wrong. So yeah,
we'll go with herd. Yeah, it's there's these things all bias. So the most, I guess, rigorous answer I can give here, most mechanistic, is phase amplitude coupling. right? So the idea that, so, we can actually go, we can revisit something from the paper, actually. How did I get these results in monkey electrophysiology, these macaque ECOG results? I used gamma activity. I used the envelope of gamma activity. So gamma activity, this is 40 to 100 hertz activity. This is presumably, most people buy that this is basically reflecting population levels, multi-unit activity, firing activity in the brain, picked up through electrocorticography. What I'm measuring here is the amplitude, the overall, amount of multi-unit activity. That amplitude envelope fluctuates over tens of seconds. So, in a very oversimplified interpretation of this, you could think about it as like the amount of, your total brain power, like how much you're thinking obviously this is an extreme abstraction, but how much you're thinking, the extent of your thinking is fluctuating more slowly, even though the quality of those thoughts, the specific nature, the specific contents of those thoughts is fluctuating much faster. It's changing much more rapidly. That's kind of how I think about it. And so, this analysis wouldn't have worked if these phase amplitude coupling relationships didn't exist. So I'm looking at high frequency activity here, but I'm looking at its slower fluctuations in its overall envelope. And the fact that slower fluctuations also seem to obey their own spatiotemporal laws, dynamical laws, seems to be, indicating that there's something meaningful going on here at these very low frequencies in terms of coordinating brain-wide activity and organizing, things that are occurring at much faster timescales. I think language also provides an awesome example of this too. Just thinking about the hierarchy of, phonemes to syllables, words, sentences, etc. And a specific packet, a specific word means very little outside of the context of a sentence. Means very, very little, outside the context of a more fully fleshed out thought. And all these things, your interpretation of these smaller units can be completely different depending on things that are unfolding over much slower timescales. The overall timescale of you reading a book, for example, that completely changes the context of all the words you're interpreting. So I think language provides a good example as well for this sort of thing.
No,
but I like the herd of
this.
porpoise. The
So
porpoise herd.
I want to ask Johan whether what Steve would say. So Johan, used to work with Steve Grossberg, who's thought a lot about kind of generative models of nervous systems and how to get them to actually function the way that we need them to function. And I'd be curious whether Steve has a take on kind of this sort of nested timescales idea. I'm just curious.
As far as I know, I don't think so. In fact, I was going to ask Ryan if he had thought about mechanisms, and then I started working through what he might say in response to a question like that, because obviously this is way slower than any neural phenomenon, like some GABA timescale. No way, right? And then I was like, well, the breathing and the heart rate, I can kind of see that just directly having an implication and having an effect on networks, but not with a stable phase relationship. Or even a stable, it's almost like you have a fixed phase relationship that is itself modulated, right? So what's tricky is the idea that that there has to be a mechanism precisely because of your data that is maintaining this, and it can't be a slow GABA, kind of thing. It has to be something weirder than that. Like
glia appear to be strongly implicated in this sort of thing. So glial astrocyte dynamics, calcium dynamics occur on this time. And again, neuronal, you can look at calcium, neuronal calcium, and you see this these slow time scales. So neuronal processes are varying on the slow time scale, but the specific reason, so to speak, for this slow time scale, I think that probably comes down to, I think what's special about this time scale is that it's kind of an overlap an overlapping frequency window for both electrophysiological processes and metabolic processes. So metabolic processes can occur on these slow tens of second time scales. So glycolytic oscillations, you put yeast cells in a dish and they'll oscillate in their overall levels of glycolytic intermediates, and they'll synchronize with one another spontaneously, just like coupled oscillator mechanisms might predict. And I think something that's special about this infraslow, you almost want to go back. We always think of like fMRI and blood oxygen fluctuations as being like, we're using this purely as a measure to infer something about neuronal electrical activity. But it's kind of interesting to go back now, having seen that this holds up with electrophysiology, to say, this is something that's synchronizing electrical activity, calcium activity, blood oxygen fluctuations, et cetera. All these diverse physiological processes are synchronized according to this process. And so, these are a lot of different, metabolic and systemic processes. And so I think, this sort of, you couldn't have these things synchronizing, for example, in like a gamma frequency, blood oxygen fluctuations aren't fluctuating at 40 hertz. So I think there's something special about this slow time scale. And I think it's a clue that fMRI is giving us this result, that hemodynamics are synchronizing at this time scale and glial activity. Yeah.
Johan, I'm curious if you think that this phase resetting might be related to Grossbergian resetting in the network, with the vigilance resetting signal. There's a buildup of a certain category or something, and then at a certain point, there's a reset, and if it could be related to this, just speculation.
I think I was thinking along these lines, but the timescale makes it very hard to make strong claims, right? Because it's not the case that if I'm in default mode o'clock, that I can't do anything, right? It's like 0.01 seconds and 0.1 seconds is a long time, right? And I can definitely interrupt what I'm doing in that timescale. So it's much more about something like an eligibility trace, just to modulate the probabilities of certain things being slightly better and worse. And where I've read about this is on a much smaller time scale in, I think, dentate gyrus, where there's this big expansion going from entorhinal to dentate, right? And that itself gives you this nice sparsification. But there was a paper, I can't tell you the citation because I forgot it, but that the excitability and recruitability of dentate neurons varies on a relatively slow time scale. This is really useful if you want to separate neurons that initially have the same receptive field. I use receptive field in the loosest possible way. So something like that is probably happening here if it's performing a function, right? So what I'm saying is that you have a lot of redundancy in neural learning and coding, and you need to kind of generate asymmetry. So one way of generating asymmetry is to have different ensembles of neurons representing roughly the same things be marginally more and less recruitable. So
interesting.
If in the same situation, which like if we take situation to mean something that takes a few seconds or minutes even, it's almost like giving various learning cells a chance to take a snapshot. That's how I would model it. At least that's how I would start to assemble papers to justify a model of that sort, and then maybe change the model later, but that's how I would start.
I see. That's
kind of turn taking. Turn taking.
Yeah, I like that.
I would think about it. Yeah.
I like that a lot. And I especially like, even, I guess a more general interpretation. I like just this idea, and I very much agree with this idea that you're kind of, it's this dynamic probabilistic weighting of different, different functions, or here, we're talking about different functional systems, but different large scale functional systems that, I'm kind of thinking about it in terms of, enhanced gradually, enhancing excitability, decreasing excitability in some probabilistic way. And it's not to say that, you're not going to be able to do anything if you're in your default mode or something like that. But we know from, decades of neuroimaging literature that you're in your default mode network, you're probably not doing very well on whatever cognitive task you're doing at the moment, but you can still do it, but so
it depends on the task, right?
Oh, right. That's a good point, Mac. That's a fair point.
I remember some Tononi data he presented at SFN some years ago about how I think motor areas fell into sleep-like states, and you could correlate those sleep-like states with errors. So even though we're talking about these marginal differences in probability, the dark side of it we all experience. The brain fart, where those. You mentioned language earlier, and there's that wonderful example of completely blanking on a word that you use regularly. Or
tip of the tongue states,
and so it's definitely something that you can align at least with subjective phenomenology.
Yeah.
So there's a lot here.
Yeah.
So I wonder if along those lines, Ryan, if you've thought about trying to run some of these in different disease cohorts, like, does it where arousal pathology sort of starts to accumulate, things like Alzheimer's disease, where the cholinergic system's getting impaired, or the LC or something, and you say, all right, how does this, how do the different waves propagate differently? That could be kind of neat.
Have I thought about it? Yes. Have I been able to do it? No. I think this is one of those things where I really think it's a project waiting to happen. I'd be very happy if anyone feels motivated to do that sort of thing. I finished up my PhD work and kind of branching out a little bit from this type of research question. And so, I'm in Mark Rakel's group, the labs. I was the last student. We're all done. So, I'd be very happy if anyone was interested in that sort of thing.
That's a sad thought in its own right, given Marcus's impact on the field, but yeah.
It truly is. And so, I think those are the types of things that I find very interesting, going through. And, I didn't realize really until getting into the weeds of this project how much, arousal nuclei are very strongly implicated, of course, in basically every neurodegenerative disease. Clearly basal forebrain, locus coeruleus, some of the first spots to show neurodegeneration. But what I didn't know is that even, in the psychiatry literature, and I don't mean to, if I'm getting this wrong, someone can happily call me out on it. But my understanding is that things like arousal are becoming increasingly discussed in terms of, different psychiatric symptoms and such, and things are thought of in terms of like under arousal, over arousal, that sort of thing. And ADHD is just one example. And so, we have all these different terms for it historically, attention and that sort of thing, but these all are, it's hard to imagine those things being kind of independent of this process. And so I think this also, there's also converging literature. I think there's a converging realization in the neuroimaging literature that a lot of the functional connectivity differences across clinical groups seem to recapitulate this basic distinction between aroused subjects and non-aroused, basically sleep deprived subjects versus non-sleep deprived subjects. There's this general topography of changes associated with sensory motor functional connectivity that you could think about in terms of, does one group just have higher amplitude waves, for example? That would be a straightforward hypothesis that I think really would hold up in some of these cases. So absolutely, I think that this is.
But you need good physiological data that's collected concurrently in order to test these hypotheses, right?
Yeah, to test it rigorously for sure. I think I'm kind of working on ways to data-driven approaches to extract this information without a separate arousal measure. The funniest thing is the best thing I've gotten so far is this is actually a supplemental figure in the paper because the paper just got too big. But if you just time lock these brain signals to spontaneous head movements, you get the same result. So it turns out that the influence of basically head motion artifacts in fMRI are probably overrated because this process is spontaneous head movements are phase locked to this process. And so you're going to have systematic fold signal correlates of head movements.
I'm so upset, Ryan, that Kevin and Alex left the talk before this comment, because I've had this exact debate with these guys and a number of other people. human brain mapping, that like there's this idea that if you can somehow just like clean the data of its noise, that you'll somehow get like this pure, beautiful signal, like you're listening to an MP3 and trying to scrub off noisy channels or something.
Yeah,
and I just think that biologically that doesn't make a lot of sense, right? These
I think you're right.
systems are all connected together in a really fundamental way, and so I'm happy to hear.
I think you're right.
Opinion.
Matt, I think you've inspired me to. I'll pull this up actually. I want to show you how cleanly this comes out. This is a head movement triggered average. This here, you see clear propagation from motor areas to this singular opercular areas to back to parietal cortex and frontal cortex. So eventually landing up in here, areas of the default mode network.
This comes back to this idea that we've been looking at, and it's kind of inexplicable when you try to explain it to folks. Like just recently chatting to Johan, who has a background in kind of physics and not really kind of as embedded in the bold world, trying to explain why we've looked at zero lag correlations for so damn long. Like what is it? Just because we found something there, all of a sudden the entire field just goes, oh cool, let's just look at this one little window.
Yeah, right.
It's like there's so much stuff going on that we've just been missing. It's quite remarkable, really.
Yeah, right. I totally agree.
Ryan, we've got two hands raised. I believe Eli's hand was up first, so Gabriel, you're gonna have to wait. Eli, do you want to unmute yourself and ask your question?
Yeah, sure. Thanks for the talk, Ryan. It was really interesting. I was
Thank
just
you.
curious if you had tried to, have you dipped your toe into much of the sort of eigenmodes literature, as sparse as it may in fact be, and like contrasting like a lot of the phase relationships, particularly the fact that you're interested in talking about energetics as well, with something like a spatial eigenmode or a geometric eigenmode of the brain. Like this property exists. You've got some sort of closed surface. Loosely speaking, activity is governed by a wave equation on that surface. So things like spherical harmonics on a circle that we use in other physical systems like atomic systems can be applied to the brain. And in this case, you're talking about this sort of these phase relationships. We have like an up phase of a particular oscillation and the down phase, and having this sort of pi phase relationship or an anti-phase relationship in this instance. And in fact, like when you do something like a spatial eigenmode decomposition, you have this natural hierarchy of energetics and complexity that come free, like the uniform mode. And you start adding these modes together, and in fact, some of these signatures like the Magouli's gradients and some of these I guess the networks are a little bit of a straw man because they do thresholding and what have you. So they're throwing away this property that each region actually contributes to all modes at all times. So when you do this discretization, you throw that information away. But yeah, I was curious whether or not you'd thought about contrasting this with something like spatial eigenmodes.
Is this just for my own clarification, is this kind of referring to like the Adhd papers?
Almost. The dogfather of a lot of the eigenmode work is actually an old professor of mine, Professor Peter Robinson. But it's a shame that Kevin has actually left as well, because he's worked a bit on them. And there are a few other papers floating around. The way that Adhd does it is not quite how I like to think about it, but just starting from the surface rather than starting from something like a connectome or something like that.
Okay.
But yeah, you basically get these like nice modes out of it, and then you can contrast these large scale, particularly like things like people move more towards this now when they talk about gradients and stuff, this continuous modal decomposition. But because you can say something like that, then something like your phase relationship like really makes sense, because you're like, oh, you've just got this up and down phase of this mode that's made up of a handful of these spatial eigenmodes, and something like the arousal system's coming in and just bringing online or removing from being activated. And you can say something about energetics, because surely the brain is like the short answer is, if you have a geometry and you have something like a wave equation that's at play, like neurons are loosely speaking pretty randomly connected, activity can propagate, so the wave equation holds at some scale, and therefore these sort of modes exist. And it's a question that remains to be demonstrated whether or not the biology actually exploits it. I mean, it's certainly got a fight in that domain. Like there is this energetic hierarchy. Like it is the case, it's easier to bring online a uniform mode or a sort of really spatially simple mode than it is to activate some just a particular region and no other regions. just from the physics, and then what
so basically scaling with like spatial frequency.
Yeah, and then the biology is obviously either going to exploit that fact or not. It seems like a good way to sort of bridge into what you're finding here.
Yeah, absolutely. To answer your question, no, I haven't played around much with that. I think it would, presumably that would tie in very nicely to this work and could extend it. One of the things that we tried to emphasize with this work, it was basically that, we tried to kind of present a collection of analyses that illustrate, kind of comprehensively characterize this process from a lot of different angles. And the implicit message here is that basically there's a lot of properties that if we were to use standard linear, and I don't know specifically about the spherical decomposition that you're referring to, but a lot of the linear spatiotemporal decompositions, basically they would fail on this sort of process due to things like interregional delays, the timescale invariance, that nonlinearity, that feature of it, the broadband frequency content. So if you were trying to do something, if you were trying to decompose the like coherency spectrum across all these brain regions, you'd have to choose a particular frequency, that sort of thing, particular temporal frequency. So that was the challenge that we were, and there's no resolution to this, right? Like we haven't come up with any solution to this or anything, but that's kind of where the state of the art is really in data analysis is, traveling waves are kind of this outstanding problem because they pose a problem to, you can't do your standard techniques for separation in the temporal or spatial domains. And then to add to that, there's this nonlinear feature of these traveling waves where they have this phase consistency across a broad frequency range, and they exist across a broad frequency range. And so basically all of these features kind of, there's a lot of literature on using diverse spatiotemporal decompositions to argue for multiple components in the data. The approach we took here was to say, kind of an Occam's razor argument, like, look, we've described this, this one physiological process that is very spatiotemporally rich and could give rise to a lot of these things that are conceptualized as different components in the data. But it's by no means, this would certainly be improved upon if there were a good way to spatiotemporally decompose the data and get out as, one component, this process, and then look what's underneath. And I think that's where the real challenge is. So thanks for bringing that up, though.
Gabriel, do you want to ask your question? We might, for the sake of time and Ryan's energy levels, so he doesn't flip back into his default mode cycle, we might make this the last question, and then we can chase up Ryan later via email if we have any other questions. So
yeah, please do. This is fun. I love hearing from you. So
and first of all, awesome work, mate. Like, I would dream to do something like this with at the end of my PhD in my second year. So I hope that I will be able to do something this cool.
Not in my lab, Gabriel. You're gonna have to go work with Mark Rakel. You have to convince him to unretire. You got no hope.
I appreciate it. I'm sure you're gonna. Yeah.
Okay, but going to my question, it's more specific about the pupil diameter stuff because it's what I work, like correlating pupil diameter as a marker of locus coeruleus activity, and also correlated with fMRI and graph theoretical analysis. So in my experience, the low frequency pupil diameter has been really difficult to correlate with anything related to the task or to performance, and I believe that it's because the pupil diameter has many inputs or is controlled by not only the locus coeruleus but also with supracolliculus or maybe it's also the rexinergic system or the hypothalamus. But in between like 0.1 hertz until 4 hertz, it's when I find like the evoked activity that I had been able to correlate it with performance, and in the bibliography had been related to locus coeruleus phase activity. So my question is, have you seen something like this in those fast fast scale in the pupil diameter, like using like maybe running a bandpass of the pupil diameter between 0.01 hertz until 4 hertz to see like not the slow trend changing the pupil diameter, but more like the fast peaks of the pupil diameter, and see if you can find this wave. Like maybe I'm asking that because I would think that that's more related with a phasic neuroenergy kind of activity, fast activity.
Are you working with human neuroimaging with fMRI?
Yes, I have.
Oh, so I generally, and this is just because I'm stuck in my ways, and I haven't. I generally don't look at above 0.1 hertz. I do buy that there's meaningful signal above that, but the SNR drops off very quickly above 0.1 hertz, and so just by convention, I've kind of, and there's different, the literature that does exist on the infraslow oscillation and the electrophysiology and such seems to indicate that much of the interesting features associated with this specific physiological process that tends to drop off. Most of it's below 0.1 hertz. So there hasn't been much motivation from that perspective to push it higher and to look at higher frequencies. And the possibility is that this does extend for a little bit beyond 0.1 hertz, but your ability to see it with neuroimaging data might decline after that. I know I'm looking at this with like mouse calcium imaging as well, mouse wide field calcium imaging. There you can get higher frequencies. You definitely see this process going faster than 0.1 hertz, but honestly, above that, it does seem to break down pretty quickly. You seem to get into the delta frequency range, and that seems to be very, in my experience, it looks quite different phenomenologically.
Yeah, no, I maybe I got it wrong. I was thinking not in the fMRI frequency, more in the pupil diameter frequency. Like,
oh, sure, but I guess in studying pupil to brain relationships, I focused on because otherwise, if I'm looking at very
using the same
signals,
frequency.
Yeah,
exactly. That makes sense.
But I think, what you're getting at is absolutely, you're totally right. There's people have talked about this tonic and phasic pupil fluctuation, and much of the people can respond very quickly, and it has much faster responses, and it's unclear actually if to me whether the immediate responses of, for example, neuromodulator systems or immediate correlates of that evoked pupil response, that fast pupil response, if that is intimately tied to this process, if that's something fundamentally different, that's a totally open question. So to answer your question, no, I'm not sure. I haven't looked into that very regularly.
Yeah, and maybe the other question related to this is what if you have a hypothesis of some specific mechanism, like, because I would think that is the brainstem and is the locus ceruleus and the hypothalamus, maybe the synergic system doing some waves and something interplay with that, and maybe is the basal forebrain and the locus ceruleus send the projection to the thalamus, and then the thalamus is the one coordinating the cortex, and that's why we find this linear trend, beautiful trend in the thalamus, but not so linear in the cortex. Or I don't know, because if that is the case, you could use maybe receptors like the data that is in the Allen Institute and see how match with some of these waves or maps that you showed here. Like I would think that maybe some of the receptor maps will match with this, and we can think that is maybe because of some neuromodulatory system having a bigger role or lesser role in this dynamic.
Just by looking at some of these maps, they do match quite well. I think you can make it just so story in many different ways, but I think that's also the challenge, is that you guys are obviously familiar with the issue of spatial autocorrelation, and you go in and look at the first principal component, right, of this Allen microarray data, and it recapitulates this sensory motor to association axis. So it's like you could pick your favorite gene at that point and say that this is related to that process or whatever. And, you know, obviously specific neuromodulator systems do have some differences and such, and you can map that out. But I just, because this is such a sweeping process, it seems to manifest in so many different physiological signals. We know that, you know, locus, like cholinergic activity, noradrenergic activity, and even, you know, other people have talked about serotonergic activity too, all correlates closely with pupil and arousal. And so I've just, because I don't have, you know, the experimental skill set to really get at these questions and disambiguate these processes. So I've kind of shied away from interpreting it, giving it that specific a biological interpretation. You know, I would love to see that followed up upon, but yeah, I think it's just,
that could be like,
there's so much involved.
Like the next, the sequence, the sequel.
Yeah, right.
Something like that.
We're better to ask that question than precisely where you seem to be spending half of your time now. I mean, you need precision data that you can then develop predictions about how to manipulate the circuitry and turn it on or turn it off or augment it or diminish it. So yeah, I think you're in a great place to push this forward.
I think so. I think they're a little bit, I don't know if you know anything about the Allen Institute, they're very like, I mean, they run like they're like a company, right? They have their operation. They don't have the kind of flexibility to pop in and, you know, be like, hey, this would be a really cool thing to test. Let's collaborate on this fun one-off experiment. And it's kind of like, actually, we have, you know, this 10-year plan that we're sticking to a T. And, you know, so from that perspective, it's a little tricky to do something like that with experimental collaborations. But
you can just use the sort of existing data for a different means
to make a strong case.
The open scope idea, I think, is such a cool concept.
Yeah, right. Yeah, I totally agree.
Have you heard about this with Johan, Kayla, and others, where they have this thing they call the open scope, where they're going to be designing experiments to be in a particular location, let's say the visual cortex, and they're going to have Cree lines that define IT cells of different classes, let's say layer 2, layer 3. And they're like, look, we're going to be there. So if you've got like a particular task that you think would be super important or a particular kind of context that you'd like to see those recordings in, just let us know. And, you know, we'll have everyone kind of vie for that. We'll have some scientific advisors decide which is the most fruitful, and then we can collect that data for you. So it's a lot like those mega telescopes they have all around the world. They're like, well, look, we spent all this money putting them together. We don't quite know what to look at. Do you want to look at any of this stuff? And then you point it over there, you're like, oh, look, that's star formation. It's super interesting to me. So I think it's such a cool concept that we need to kind of embrace, I think, a bit more as a field, because we'll be able to answer so many questions with this kind of data. It's just about being able to ask the right ones to kind of really test between different hypotheses.
Right.
Cool. Well, as always, SNAC has ended on me rambling about something in neuroscience. So Ryan, that was awesome. Thank you so much for a really lovely talk, and it's really great to kind of see, you know, under the hood, to see how a lot of this sort of went together and kind of the ideas you were thinking about. I'm really curious to kind of see how this kind of expands out into the fields of both kind of, you know, task related functionality, but also in different disease cohorts, as you mentioned, psychiatry, but also neurodegeneration. I think there's a lot of really interesting questions to think about in this space. So. Thanks
so much for having me. This was, I really appreciate how, you know, engaged you all were. This was probably the most fun presentation I've given. It's everyone kind of asking really great questions and just being very, very involved. So I really appreciate that.
Awesome.
Please do feel free to reach out after. I'd love to hear from anyone too about further discussion. So
cool. All right. Well, thanks, man.
All right. Thank you all.
Take care. See ya.
Thanks.